And we're live.
Hello, everybody.
Hello, everybody. Now type, Scott. Let's type something.
Hold on, let me put it. Let me pull up the
No,
private
no.
chat. I'm tapping.
Okay, very good. Okay,
All right.
that's enough. Yeah,
Okay.
quit now. Quit now.
Okay. I got a question for you, real quick, before we get into all these heavy brain things. Have you seen the any of the trailers or advertisements or interviews for um, Project Hail Mary?
No.
Now, it's not like a religion thing. Of course, I don't know. There may be some religion in the movie, but yeah, it's Andy Weir who wrote The Martian, and then they did that movie The Martian.
Yeah.
Well, he wrote another book that was wildly successful, and they've made it into another movie. I just wondered if you'd seen any of that. It looks pretty exciting.
Okay.
Dr. Becky Smethurst
Okay.
did an interview with many of the people that behind it, like Ryan Gosling, who plays the main character, and the creative team. I think she aired a previous interview with Andy Weir, but anyway, it was all pretty exciting stuff. Whatever the marketing team did kind of hyped me up as well. So,
Okay.
but it's kind of Andy Weir's one unrealistic thing is that one guy could do everything in space and live.
Yeah.
That's the um, what's it called when the something of disbelief. Where you set aside the temporary suspension of disbelief. That's really what I gotta say is the thing that makes it hard to believe. In my view, um, a single astronaut going out into space by themselves for a long time, whether it be to Mars or to live on Mars or to go to what Tau Ceti, I think maybe that's the star they're going to. Um, that just doesn't seem realistic. He'd be a fried husk of a irradiated person after a certain amount of time. I'm afraid because you just can't do it all. You got to have a team. But
The
that's my
real
own
problem.
opinion.
Yeah, the real problem with aliens is that they're too damn far away. Anyway, okay. So I talked a lot about zygotes. Trying to make the point that a zygote is one damn cell, and you are one cell to begin with. And how does one cell turn into a complex human being with a brain, liver, kidneys, heart, all of that stuff?
Yep.
And that's sort of a big story. And this also leads into talk about the Ediacaran and the Cambrian explosion, and a point that I was trying to make about gene regulatory networks and systems, basically. But all of that kind of relies on having an idea about embryology. And when we talk to people like Jim Nub, and we're always telling them if you understood embryology, you would understand evolution. And embryology is the key to evolution. There's also Haeckel's drawings and ontogeny recapitulates phylogeny.
Right.
Well, it kind of does. It kind of does. When you look at how an embryo develops, basically the same patterns, exist in the frog as in the human, the mice, but there are differences. There are very early differences that are like a butterfly flapping its wings. The other thing I talked about is I'm trying to use my campfire analogy to understand how genes are information. Genes are information almost strictly only to create a particular protein. When you say that a gene is information to create an organism, you're kind of lying to yourself. It's not like that. It would be like if you had a campfire and it burned down, you had a whole bunch of embers in there, and you moved the embers around, and moving the embers around changed the smoke that was wafting off the campfire. Okay. That's how genes work. So if an ember is in one place, it's going to create one kind of a smoke column, and you move it a little bit and it's going to change that significantly. And it's almost like a butterfly flapping its wings. So those are the reasons why I like to talk about embryology. Now I found a site here. What is it called? It's called Alila Medical Media, and it has a whole bunch of really good animations. And I thought I'd play one, but they have the most goddamn irritating intro music I have ever heard in my life. But let's go ahead and play this, and we'll see where this goes.
Anyway, because you might get a copyright strike.
Well, we'll see. I don't know.
Live dangerously.
Who gives a shit? Okay. So
before you get started, I wanted to I wanted to mention a Anton Petrov video that was really fascinating, and that is, it was from, when was it? From a day ago. We were wrong about the human appendix for over 150 years. I'll put the link in the side chat, but it was pretty interesting. But he says Darwin was wrong or whatever, so I know the creationists are going to love this, but it's fascinating, and it has to do with management of gut bacteria.
Microbiome, yeah. Yep, okay. Yeah, go ahead and create a link for me so I can drop it right into our description, because if we mention a video, we have to put it in the description. That's a rule. Resist depression, we got Ryan Murphy, and we have laborious cretin. Genes get to predispositions. It's not 100% due to feedbacks and epigenetics and biochemistry. Not sure what you mean here. It's not 100% due to feedbacks and epigenetics and bio. Could be a little clearer about that one, laborious screen. Anyway, okay, we're going to go ahead and play this now. Quit typing.
That's a strange background music.
That's their intro music. They have it on every video, and it's like, that's the most aggravating thing I've ever heard in my life.
Human prenatal development can be divided into 3 stages: pre-embryonic, embryonic, and fetal development. Fertilization usually occurs in the ampulla of fallopian tube. The fertilized egg, referred to as the conceptus at this stage, immediately travels toward the uterus. During the journey, it undergoes several mitotic cell divisions, producing daughter cells called blastomeres. This process is known as cleavage because the cells divide without growing in volume. After about 3 days, the conceptus arrives in the uterus. It now contains about 16 cells and is called a morula. Cells of...
I just want to interject something here. In the mouse, by the time it arrives, it has, I think, 24 cells or 32 cells, I'm not sure. But there's a very slight difference in every animal as to how many cells are going to be in the morula, which is a mulberry. Okay, it looks like a mulberry. So that's you, basically, when you have 16 cells. You are just a mulberry-looking thing. What?
It says sweet 16.
Sweet 16, okay.
The morula are totipotent. They are capable of differentiating.
I always pronounce that totipotent, but totipotent is harder to say. Anyway, totipotent means it can create any cell, including the embryo and all of the placental and extra embryonic tissues. So it pluripotent means it can create anything in the embryo. Multipotent means it can create anything in a particular organ class. So now it's gotten down. You can imagine we have an onion, and or a set, and inside that set, so we have totipotent, multipotent, or pluripotent, you would pronounce it, and then multipotent. Basically, goes all the way down.
Well, see, I've only ever heard the word pronounced pluripotent. So
yeah,
I don't know if there
so,
are some unexplored rules of English accents
maybe
that
AI
are
is reading this.
maybe AI. Yeah, because I love it when the word is supposed to be live, like you know,
And they do live. Yeah.
live and they call it live.
Yeah. Yep. Okay, we were wrong about the appendix. So there's Scott's thing. Okay. Now, how can I copy that thing?
No, don't copy that one because I had to delete it because I actually put the wrong link in there. The title's right.
Go ahead.
So I'm doing it.
Okay, so we got a fertilized egg. So somebody had sex and it's moving down. You saw that that little place where they had two nuclei. That was the daddy nuclei and the mommy nuclei. Okay, and they go down. Okay.
Into all cell types, both embryonic and extraembryonic. The morula continues to divide while floating freely in the uterus for several more days. During this time, it consumes nutrients stored in the egg cytoplasm and uterine milk secreted by the endometrium. When there are about 100 cells, the cells start to arrange themselves around a fluid-filled cavity forming a blastocyst. The blastocyst consists of pluripotent inner cell mass called the embryoblast.
And that's you. That little part in there is, well, part of that is you. There's, we're going to shed some more. So it's almost like we have an eggshell and we shed the eggshell.
Destined to be the embryo, and an outer shell called the trophoblast, which nourishes the embryo. The trophoblast later becomes the chorion, the fetal portion of the placenta. The trophoblast secretes an enzyme that dissolves the membrane surrounding the conceptus, enabling it to hatch, ready to be implanted. At the end of the first week, the blastocyst becomes attached to the endometrium. At the site of contact, superficial cells of
Now, this is where the parasitic nature of the embryo becomes obvious, because it's going to stick fingers in there and it's going to invade the uterus and it's going to start to take over the female body.
So if we
including
filmed it, like videoed it real close up with one of those microscopic lenses or whatever, it would look like a sci-fi horror movie.
Yeah. What do we got here? Let's see. Second, I'll expand small spaces too tight. We got, let's see, genetics for height being one. People can get feedback during growth. That sounds growth. Yep. Yeah, it's complicated. Anyway, okay, so let's go a little further with this. This is kind of the 10,000-foot overview.
Of the trophoblast fuse together to form the syncytial trophoblast, which grows into the endometrium and derives nutrition from it. The endometrium responds by growing over and eventually enveloping the blastocyst. The syncytial trophoblast secretes the hormone named human chorionic gonadotropin, HCG, to instruct the corpus luteum to continue the production of progesterone. Progesterone is the hormone that maintains pregnancy. Among its many functions, progesterone stimulates the growth of nutrient-rich decidual cells to feed the early embryo. Around the middle of week 2, the embryoblast starts to form a two-layered embryonic disc, hypoblast and epiblast, at the midplane.
So now we have a bilaminar disc, basically, and two-layered. But it's interesting, the hypoblast becomes yolk and a few other things. The epiblast is you, so you're that little thin blue line, tiny blue dot, thin blue line, whatever. Oh, I think you can actually see my cursor. Yeah, you can, sort of, right there. So that's you at that point, and the rest of the you is being shed off into whatever it is that gets you born.
Of blastocyst, with a cavity on either side. This bilaminar disc sets the dorsal ventral axis of the embryo, with the epiblast being the dorsal.
So that's your back and your front. So basically that axis is set up early on.
Dorsal side. By the end of the second week, The two layers grow around the cavity on their respective side and form the yolk sac and amnion, respectively. The yolk sac provides nutrients absorbed from the trophoblast as well as gas exchange for the embryo during weeks 2 and 3 before the placenta takes over these functions at week 4. It is also the first site of embryonic blood cell production before the liver. In week 3, the two-layered disc transforms into 3 germ layers through the process of gastrulation, during which the cells lose their pluripotency and become committed to a smaller number of cell types. Gastrulation begins with the emergence of the primitive.
Okay, this bilaminar, trilaminar. Some animals remain bilaminar, like some simple, I think, sponges. I'm not sure exactly, but there are some very simple organisms that basically have a simpler development path than we do as mammals. And that's an important branching point in evolution. And we'll hopefully I'll mention something about that a little bit later.
Primitive streak on epiblast surface. This event sets the cranial caudal or head-tail axis of the embryo. A so-called
that primitive streak is the beginning of your nervous system, and it comes from the outside or the skin, not the endoderm, but the what is it called? Can't think of the name.
Primitive node at the cranial end of primitive streak produces signaling factors that control the movement of cells. Epiblast cells migrate toward and through the primitive streak into the embryo. The first set of cells displaces the hypoblast and forms the endoderm. The second set of cells fills in
that's inner skin,
the space between the endoderm and epiblast, creating the mesoderm. The remaining epiblast cells form the ectoderm.
So that's the outside of you, basically, ectoderm, mesoderm, which differentiates into a whole lot of things, and then the endoderm, which is the inside skin, sort of.
Each of the 3 germ layers differentiates to become different organs and tissues. All organ systems are present in their primitive form by the end of the embryonic period, but most of them are not yet functional or only partly functional. The yolk sac gives rise to the allantois, a primitive urinary bladder and part of the umbilical cord.
Okay, now here you can see basically the yolk sac is kind of continuous with the inside of your gut. And there are branches from that. And one of the allantois basically is a primitive, like that's the beginning of your urinary system. But for the embryo, that collects waste. Okay, so the yolk sac is input, it feeds you a little bit, and the allantois takes away the information. But then later on, these branching points become liver and pancreas.
The amnion grows to surround the embryo by week 4. It is filled with amniotic fluid, which protects the embryo from trauma and temperature fluctuations. The fluid also allows the fetus to move freely and prevent adhesion.
Okay, at about 28 days, they transfer from calling it they transfer from calling it an embryo to a fetus. So fetus after, the first month, about the 30th day. That's when they'll, and this picture with the little eye, I don't know, that's very cartoonish. And that creates problems for, arguments about basically abortion.
Of body parts. The yolk sac, amnion, allantois, and chorion are the 4 embryonic membranes.
Let's discuss this thing right here that looks like an eye. What's that supposed to be? Because it starts out with a kind of a white fluid-looking thing, and then please go ahead.
Okay, I'm not sure what your question is.
Oh,
okay, the eye.
See between my YouTube video and the StreamYard video.
Oh,
but so okay. They weren't synced up. But you said that there was a problem with the eye. Are you talking about the embryo's little dot here, the little gray dot?
Yeah, the cartoon that they drew is what I have a problem with. They got little tiny feet and stuff,
Uh-huh.
and I don't think that's how it really looks. So I kind of wish they had drawn it a little more realistically,
Yeah,
so you have an
because
idea.
so I
Yeah.
wonder who if there was a motivation to draw it like that so that they wouldn't get screamed at by right wing groups.
I don't know. I'm not sure, but I wish they would. Well, I got some pictures later to show you some of the stuff with the brain and things. So anyway,
or
okay,
embryonic membranes. Are you a fan of Alila Medical videos?
that's it. Okay, so we're done with that. Let me get rid of this.
So is Alila Medical Videos primarily embryology, or do they have a lot of other?
A lot of other medical stuff, a whole lot of medical stuff. It's really great. I linked their site in the description for anybody that wants to. Yeah, Levin. Michael Levine or whatever touches on, but he's trying to run into the deep end also. Yeah, there's there's all kinds of bioelectric things going on in the cell because you have sodium and potassium and calcium. You have ions basically, and that is going to create some, and chlorine, of course. So that's going to create electrical fields. And Michael Levin has taken that and he has been doing Essentia videos and he has just kind of gone nuts on this stuff. It's He's a brilliant, brilliant researcher, and he's done some really brilliant stuff on cell development that digs a layer deeper. But other than that, don't listen to the guy. He's kind of gone off the deep end. I have no idea why he would do that. Maybe he had some religious background. I'm not sure, but
Now, how
his
has he
work
gone
is
off the deep
still.
end? He's putting
What's
feet
that?
and eyes on. He's gone off the deep end by putting feet and eyes at the wrong stage on his pictures, or
No,
uh,
no, no,
other
no.
things.
That video had nothing to do with Michael Levin. Levine, I think it is. Michael Levine is a researcher who discovered a whole lot of stuff on bioelectric fields back in the early 2000s, I think, and he got a whole lot of pushback on that. Everybody was saying, that's woo shit, So he was a little butthurt from that, but then it came out his research was really powerful. And he's the one that created, like, he took some frog cells and he ended up putting them in a different environment, and they ended up growing into little tiny animals that did things all by themselves. And
What?
but then, yeah,
So he
yeah,
took tadpoles and put them in,
we
or
could
before tadpole?
before tadpoles,
Huh.
and yeah, he took little pieces. He's also working on limb regeneration and stuff like that. So he is really at the cutting edge of all this stuff. But anyway, that's kind of a side thing. So let's see. Keeps running into religious beliefs that don't pan out. Yeah, it's pretty bad, some of the stuff he does. Anyway, okay, so we got that. So I'm not sure. Can you see that? See the video? So anyway, we have fertilization, and we have the two, pronuclei, and they get together.
Is there a
And
way
then
you
we
can
have.
enlarge it on your screen?
Yeah, I can make it a little bit bigger, I think. Let me see. Oh, Christ. Yeah, I can make it bigger like that. Is that better?
Yeah, it's a lot better.
Okay. And then then goes to cleavage, and it's really interesting because these cells, they have a polarity, sort of, some of them, and some of them, they have a north and south pole, and so they have a top and a bottom, and they will divide, and the bottom part ends up not having a north and south pole, so that ends up sticking in the center of the mass, and then the outer part ends up becoming this hypoblast later on, and the inner cell mass, which is right here, is the embryo proper, basically, with one caveat we'll get into later. So that's the inner cell mass. And the other thing, these polar cells on the outside, they're very interesting because they suck in water, basically, and they deliver water into the center. And that's what creates this huge space. And then other cell mechanisms cause this inner cell mass to stick together. It likes to kind of, it's a little bit sticky, and it gets pushed aside into one side, and that ends up kind of setting up your front and back and your embryo, basically, at this real, just a few cell stage. And that little sliver of cells is you. This is you, and you're going to shed even more of you a little bit later. So then what happens is this outer cell mass divides into two different things. There's something called the rule of pairs, where almost everything divides into two things at some point. The outer cell mass divides into that syncytiotrophoblaст, which is what embeds itself into the uterus. And then the cytotrophoblast is the inner part that kind of like goes on to develop more structures. So, and then we end up down at this stage where these things start to form their own little inner cavities, right? And we got the embryo, let's see, bilaminar disc, I think. We see, yeah, okay. Yeah, the epiblast right here ends up with its own little cavity. Well, we've jumped ahead. Okay, here's the inner cell mass, and it goes ahead and it embeds itself. And then the syncytiotrophoblaст starts to develop. Syncytio means the cells lose their walls, and so we have one big mass. It's just a bunch of nuclei, and that happens in flies and a number of other things in really remarkable ways, and create different kinds of organisms. And this is the butterfly flapping its wings. The things that happen at this early stage. How many cells are involved in here? In the mouse, we have more cells at this stage, and in humans less. And there's all sorts of interesting things that happen. But this syncytiotrophoblast, that mechanism also works on the embryo of the fruit fly and creates, forgot the name again. All these terms are very hard to keep in my old mind. They end up being what the outside of the embryo. So the same mechanism that creates the thing that embeds itself in the uterus in the fly has been commandeered to do something entirely different. And we'll get into fly development at some later point, hopefully, if people like this sort of thing, that sort of thing. I don't know if people like this. Opisthecont. Archontostolictin, Sarcopterygian, Haplorhine, Symbiform, Catarrhine, primate brothers and sisters in the genus of Homo. There you go, Austin. That's a good one. Yeah, xenobots is what Michael Levin was talking about. Anyway, that's another interesting thing that we could address at some point. Okay,
yeah, here's something. I did a Google of Michael Levin frogs, and
yeah.
I haven't explored it further, but he talked about manipulating the bioelectric signals or ion channels between cells. The team showed that cells can be instructed to build organs like eyes in abnormal locations, which is very monstrous, but they showed that even if facial organs start in the wrong place, Picasso tadpoles, the cells work together to rearrange them into a functional normal face. Now get this, suggesting a high-level intelligence-driven morphogenetic process.
Yeah, but it's not.
That sounds a little weird.
Yeah, that's where he goes off the rails. He's starting to think teleology and stuff, and it's just it isn't like that. And hopefully we can illuminate a little bit of that here. Okay, so then we go to the bilaminar stage right here. So we have a bilaminar disc, which is the hypoblast and the endoblast, and or epiblast. The epiblast becomes, let's see. Now, I guess okay, there's a trophoblast, the hypoblast. Hypoblast is the brown stuff. That ends up becoming the yolk sac, basically. And the epiblast, the bottom part here, ends up becoming you, the top part ends up becoming the amnion, which surrounds you in the uterus, basically. So there again, you shed more of you, and now you is just this little line right here. This little single layered bunch of cells or mass of cells, and then it divides in two. And they don't show bilaminar disc formation very well. But anyway, these cells end up becoming, the top ones are in one environment, the bottom ones are in another environment, and it ends up becoming a bilaminar disc. And then what happens is the thing starts to fold inward so that some of these cells from the outside end up going into the inside and they form a trilaminar disc. So that becomes the mesoderm, which ends up developing into a whole lot of stuff. And until I get the actual lineage down, I don't want to talk too much about what it is, but muscles and somites and things like that. But now this primitive streak forms, and it's very interesting. It forms like there's basically a field of different proteins and bioelectric fields and things like that that create this streak. And this we have a picture of the streak, which we can get into later. But that ends up being parts of that end up becoming your brain and nervous system. They come in from the outer skin, the epidermis, and fold inward, and some of them become mesoderm, some of them end up becoming your brain. And then let's see, we got yolk sac right here, and they have two layers, and then we have the exocoelom, which I believe starts becoming your digestive system. No, that's actually here. That becomes your digestive system. Anyway, so now these little guys, the syncytiotrophoblast, has embedded in the uterus. It's got the yolk creating some blood cells, some initial blood cells, and we're getting started basically. And this little guy that's starting to curve around is the embryo, and the outer part of that is going to go all the way around and cover the whole thing and lock it in. So anyway, let me see if I can go back a layer.
I wonder what would happen if I to my 6-year-old grandson.
If you what?
If I showed that chart to my 6-year-old grandson and say, "This is you, in my tummy," and see how he would react.
Yeah. Oh, let's see. I've got another picture over here on the left that shows. There's a lot of complications. Okay, here's the inner cell mass and the trophoblast, which ends up sticking into the uterus and stuff. But the reason I captured that picture is because I wanted to show you something about cells. They have these membrane channels, and some of them actually punch right through from one cell to another, and they create this hydrophilic channel, which all kinds of information can pass through. And let's see here. Oh, if you want to see what it really looks like, that's an inner cell mass forming, or this is a morula. Basically, that's some of these cells are going to become you, some are going to become other things like uterus or not uterus, but placenta, stuff like that. Oh, let's see here. Okay, 12 to 32. Okay. I thought this was useful for, let's see. We got the amniotic cavity, and we got you developing, and here's your primitive streak where your brain is going to start developing, and this embryo is the. Excuse me.
So it looks like a bunch of mushrooms.
It does. It does look like mushrooms.
That's why you like it.
That's why I like it. Yep. And this blue part, ends up becoming you. And then another look, this is a cross-section look down here. Let's see if I can make that one bigger. Come on. Oh, no, it won't get any bigger. It don't get any bigger than that. Okay, that primitive, this little blue part here is the brain. And we got the yolk sac, and, becomes your gut and all of that. So, and let's see if I can go back another one. Okay, am I on day one? About 4 to 6 week embryo. Yeah, okay. Yeah, so this is just more of the same. And then these become a little bit hard to see, and I can't go any bigger, unfortunately. But this shows, the first week, and it shows a division of. You end up with 2 yolk sacs. You have a basically a primitive yolk sac and a final yolk sac, and there's just all sorts of things going on. And this is the primitive streak, which ends up becoming your nervous system. And this is another view of it right here. This is kind of the embryo with the bilaminar disc, and that primitive streak is creating the middle of you, the mesoderm. And here's another cross section of it, where we have mesoderm and we have neural development on that side. And then let's see, on week 3, okay, the embryo starts to take on its, it starts folding inward. This is the head right here, and folds inward, and then you got the butt and the tail and all of that. And there's some cross sections. And these are electron micrographs of what you look like. That's a fetus. Somewhere in there is a soul. That's where the soul gets imprinted. So.
Oh, they say that, huh?
Yeah. No, not in my books. They don't. But anyway, and these are some pretty good drawings of the embryo forming, and here's like a primitive limb bud forming the head.
We have an inquiry from Austin Sims. It's on the screen.
Okay. Starts off as a oocyte, and then it gets fertilized and becomes a zygote, and then it becomes an embryo, and then a fetus. Yeah, you got the order kind of correct. But not a blastopore. It's blastomeres are what they call the cells in that inner cell mass or whatever. Okay, and then here's some actual pictures of. Now, this is really interesting because this is your brain right here.
Yeah, that looks like a monster from a sci-fi movie.
Yeah, it does. Okay. You see these. So this primitive streak is very messy. It isn't, they draw it like a nice little line, but it's kind of a messy thing where it starts folding inward. And it's really neat because it folds down, it evaginates basically, and it forms these ridges up here. And then those ridges get signals where they fuse together. And this part is your nervous system. And part of that ends up being the head nervous system or the brain, and then your spinal cord, and then your butt and your tail, that sort of thing.
Yeah, I think maybe they need to early on in these videos mention that they're catching the best shot, of photograph shot. They're drawing the most well-defined picture. That in real life things are a bit hazy and mushy and harder to define. If it is, I mean, there may be cases where things are easy to define, but anyway, they need to let people know that things aren't so clear cut and clean.
Yeah, these cells basically. So each of these cells is its own little organism in its own little environment, right? And when it splits off, we got this environment, the yellow environment on one side and the green environment on the other side. And it's going to make a difference between the front and the back of you. And then other signals, some of them coming from the mommy, will end up creating the head of you and the tail of you. And this thing will slowly develop. This is showing it embedding in the amniotic cavity, or the uterine wall or endometrium. And it shows how it just sort of invades and then it sticks itself in there. And now the unfortunate female has a parasite. So, and then it starts to develop the yolk sac right here. Again, the blue part is the U that's coming out. And let's see what else we got here. Yeah, these are pictures of the primitive yolk sac and the definitive yolk sac. The primitive yolk sac gets kind of like turned into a definitive yolk sac at this stage right here. And let's see what else we got. What else we got? Formation of the chorionic villi. That's how it gets fed, basically. Let's see what other pictures I got here. Okay. Yeah, this is sort of a diagram. This is the division, like every cell, those pluripotent cells, totipotent cells divide and turn into the what is it? Trophoblast and what is the other one? Forgot. Epiblast and the epiblast and hypoderm, I think it is, or hypoblast. And then it divides again and again and again, and these cell types keep dividing and differentiating. And for complicated animals like us, there's about 20 or 30 stages of differentiating, basically from one type into another. Now, when you get to this diagram here, here's the neural tube that's formed, and you remember I showed you a picture of what the neural tube really looks like, and it joins at the top. And this becomes your nervous system, right? And then there's a notochord, which is really an interesting evolutionary leftover, sort of, this notochord. And the notochord is sending signals to one side of this brain, and the environment up here is sending different signals. Then we end up with mesoderm, and you end up with something called somites, which we can see in another picture here later on. And that ends up making you look a little bit like an insect, where you have, segments. And those segments develop into your limb buds, your arm buds, your ribs, all the different parts going on down. And that has to do with Hox genes. And remind me to tell you a little bit more about those later. But so the somatic tissue, you end up with the coelom, which ends up becoming the inside of your abdominal cavity. And then inside of that is your gut. And then you have an inside of your gut. But we have the somatic tissue. Basically, it turns into muscles and things like that. And then we have the splanchnic mesoderm. Splanchnic is a word I just love to say, even though I have trouble saying it. Splanchnic becomes your organs, like your liver. So this is the division where basically these things are going to become muscles, and these are going to become liver and pancreas and things like that, and heart, all of that. Okay. Is any of this useful?
I don't know how to answer that yet.
Yeah. Okay, and then when you really get down into it, this is the pancreatic and liver cell lineages. What we have here is...
Well, it's informative. It's just that, at my level of place in life, I don't know where I'm going to be able to repeat this back to people. You've been doing a lot of studying and explaining, and now you're doing videos about it, so that reinforces it for you. But
don't try to repeat it. Do not try. Never try to repeat it back. That's what I'm trying now, and I would have studied for another 2 years before I got really good at repeating it back. But yeah, I don't expect that.
Try to repeat it too.
I think what I'm trying to show you is this thing about the zygote, right? This little cell that has no real shape at all, and it keeps dividing and turning into other things, which go through all of these different cell types, right? And create 200 different tissues in your body by the time we're done. But what this is this shows you ventral endoderm, dorsal endoderm, and it becomes, let's see, the pancreatic bud, and that starts creating your liver, basically. And this one creates your pancreas, based on all of these different genes that get turned on and turned off in the cell. And we'll try to drill a little bit more into that. But I just wanted to show that these cell lineages are kind of like, it's interesting, but here's the mommy, and it divides into two different kinds of things, right? B cells and gamma cells and alpha cells. And, I mean, and they keep doing that all the way along. So there's this cell lineage, basically, this ancestry inside of your body, where one cell came from another, one cell type came from another cell type. And let's see. Oh, yeah, this is just more of the liver development and stuff, because I've got a bunch of stuff here on liver. Somites, okay, there's more liver, or bamboo shark, Jawless vertebrata, chordata. And I don't know why I have that one. I'm not sure. Oh, yeah, that's liver development. We'll get to that a little bit later. This is that, what is it, cephalocordate, that the Amphioxus, basically, or lancelet, and it has a primitive liver. So Jim Bob and Theus will ask you, how did the liver develop? Well, originally this thing was kind of a mutant yolk sac that came out here, right? Created this mutant yolk sac, and it ended up becoming useful because food would get in there, and there were some cells in there that helped digest the food, and then went back out into the gut and on down. And so it helped with digestion early on. And it had like it has one or two functions. The Amphioxus is a extant organism that exists right now, today, but it has a real simple liver that we can use as a model, and we can infer that early livers probably started out this way. And then when we go and look for genetic traces of that, we find out that these cells have the same gene expression as liver cells. And we find out that the method that creates this little diverticulum, what is it called? A hepatic cecum, it's called. The genes that create that little yolk thing are the same ones that create the yolk sac. So we've got evidence from genetics, because they're using the same system to create these little tiny primitive liver. The same genes are used in creating our much more complicated liver. So anyway, let's see, what do we got here? A lot of stuff's out there in spots online. Yeah. So good information, especially with how it's butchered out on the internet. Yeah. Okay. What else we got here? I think we're back at the beginning. So the next thing. Now, all of this gets down to cell development, basically. And there are many different kinds of cells, like, 200 tissue types in your body, or 200 plus tissue types in your body. And when you start, the next thing that you start getting into is cell biology. And some cells form these nice little cuboids and squares and things like that. And some cells are round, and some cells are shaped like they're narrower at the top and the bottom. But all of that has to do with what's going on inside. And all of what's going on inside has to do with what's happening in the nucleus. So we have gene expression for a certain kind of cell. And what it's going to do is it's going to create some internals that's going to determine the shape of the cell. And we have microtubules and actin and stuff like that. And then it's also going to determine what kind of cell membranes we have. So basically, it is going to give off paracrine signals, are called. So it's going to give off some proteins that go out and signal other cells. So it becomes the environment for other cells, and those other cells are going to deliver stuff back in. That stuff is going to go through this complicated regulatory network, end up inside of the nucleus, and it's going to change the nature of this cell. So that's how cells divide into two different cell types. Is that clear? So basically, every cell is like, it exists in an environment, and it's giving off signals and it's getting signals. And at some point, it locks in. It's like it starts to change what its gene expression is until it locks into a cell type. And hardly anything that's called it starts out as a stem cell, and then it becomes basically a particular cell type. And it's this feedback mechanism where it's giving off things and it's taking in things and it's going through a process, and then it locks in. And now that nucleus is only going to produce the proteins needed for your liver cell or your heart cell or your neuron or whatever. So it self-directs its own development, let's say. Has its own DNA distinct from the mother, and it's left to develop in a supported environment, follow an unbroken trajectory. Yeah, but it needs an environment too, because if you change the outside of it, and that's where Levin's bioelectric fields, and it isn't just bioelectric fields, there's a whole bunch of stuff.
I
Now,
wonder
interesting.
why the embryo self-directs its own development. I don't know what to make of that. I mean, it's a automatic. It's an automated process in the sense of, you've got all the stuff there and all the biology is going to happen, but I don't know what they're trying to imply by saying self-directs.
Well, he's trying to say that basically that zygote has everything it needs to direct its own development. Sort of. It's in a maternal environment. It's in the it's inside of that. What is it? The HL. The mother's ovum has an extra cellular network around it, right? And the mother's ovum also, there are also differences from top to bottom as to which kinds of proteins there are. And so it's going to direct it in a environmental way to start to differentiate. But then once it starts to differentiate and it gets down to that mulberry stage, basically the DNA of the zygote takes over from the maternal leftover DNA or mRNA, basically, and starts to direct its own development. So he's correct. So let's see. Personhood depends on current functional capacity, and people who are asleep in comas also lack of, and most people find unacceptable. Yeah, that's a good point. I don't know. Yeah, and the thing is, there is no brain activity in a fetus up until I'm guessing 3 months, 4 months, something like that, before you're going to get any neural firing at all. 3 months, I think. Anyway, but it's interesting. It's interesting to ponder philosophically what we were, basically. Like we were all, me and you were those little tiny cluster of snot cells, inside of that morula, at one point. Anyway. But there's a whole bunch of stuff you can find on biomechanics of cells. This is another picture, and I can't make these bigger, and I don't know why. But basically, there's just all of these things that, like this right here, it's actin, and it can constrict a cell. It can draw it together, and so you'll end up with a cell that's narrower at the top than it is at the bottom. Excuse me a second. Have to cough up a cat fur ball every now and then. Okay. And there are microtubules and all of that stuff. Now, all of this shapes a cell, but the cell is like this really complicated thing. It's got traction forces. So it has mechanical forces. You remember Ben, God, I forgot his name. I'll think of it in a second. We had him on, and he was talking about these mechanical effects on cells where they pull on proteins and they end up changing the gene regulation because of mechanical influences. So we have mechanical influences, and then we have things called cadherins that can make cells stick together. And that was really important in the evolution of multicellular animals, that these cadherins developed and started sticking cells together. And then we have things like cilia that can pop out. There's pressure, there's shear stress, cell to cell shear and normal forces, cell crowding. when cells kind of push against each other, all of these forces, mechanical and electrical and chemical forces, start differentiating these tissues in the cell. It becomes very complex from here. There's, let's see what else we got. We got a model somewhere. I don't know, they have a mechanical model. What is this? This is a mechanical model? No, this is interplay between molecular nanomachines or something like that. A picture of a neuron, which ends up being a really weird shaped cell that can be like 3 feet long, one cell, that sort of thing. So anyway, that is kind of the story of real early development. And what I want to point out is, oh my goodness, where are you? Okay, and like right around, let's see, let me get a different color. Right around the ediacaran, okay? And we got the Cambrian up here. And we ended up with, multicellularity popping up here, where cells started sticking together right here. And these cells that stuck together ended up, why is there no two-celled animals? This is bullshit, because once they start sticking together, a lot of them are going to stick together. So when a lot of cells stick together, there's these cells on the inside become in a different environment than the ones on the outside, right? And so then we start ending up with differentiation of cell types based on genetics. And these animals survived, so everything has to survive at every stage, right? And goes on to start to create organisms that maybe have like a little pocket where food can get trapped in, right? And then they might have some cilia or whatever. And the thing is that these developmental stages started out, are you with me, Scott?
I am with you.
Okay, so here at the top, we just have like one cell type, right? And then it goes down and then it ends up with an animal with two cell types and onward and onward. And this differentiation, basically the changes in the genome determine how this differentiation is going to work, right? And it's going to keep going down in layers. And what we have way out here, way out with humans, right, is we have about 20 or 30 of those layers of development. And so now that we're humans, we can never go back and become dogs, okay? Because we followed a trajectory all the way up. And this trajectory involves careful changes at each step in this inner, goes cell differentiation type thing, so that we end up with more and more cell types. So if you look at simple animals like sponges, and Austin would be better at this than I at this point, but all of these simpler animals, they have less cell types and they have less complicated genetic pathways or embryological pathways to develop. And because they have less of these things, less of these layers, they can create vastly different forms, basically. So we've got, this one animal type, which is a single cell, and it goes out and it can create many, many different body plans, basically, because it's simple. It only has one step, right? And you get 2 steps and 3 steps, and you start to get locked into these channels. And by the time you get out to humans, we have this, basically, I'm screwing my metaphors up here. We only have one thing that we can become next. We have to stay humans. We have to stay primates. We have to stay fish. We can't change into anything else because we have too much baggage going on, too much fine tuning of the genome by natural selection that we can't back up. And so in the Cambrian explosion, when all animals were of simpler types, we ended up with the 7 different body plans, basically, that ended up working. We could have ended up with 20, we could have ended up with 3, we could have ended up with 1. But once you go down that DNA pathway, it becomes a lot more difficult to differentiate, sort of thing. Oh, what do we got here? What the hell kind of stuff do I have? More pathways. Let me see. Error correction. Oh, let's see. So, and the other thing is that, you got a section of DNA, right? And they have Hox genes. And Hox genes kind of exist right next to each other. And as you go through like the segments of an insect, these Hox genes will turn on one at a time. These Hox segments, basically. So for one segment, we'll have this one on. For another one, we'll have this one on. Another one will have this one on. Well, what happened in evolution and around the Cambrian time is these Hox genes duplicated. The entire patch duplicated. And now you would only have twice as much protein, right? But then when they start to mutate, they start changing, and then they duplicate again into another different type. You end up with different packets, and what happens at each developmental stage, like the big stage, and then the inner stage, and then the inner stage, you end up with more and more complex internal organ differentiation, basically. So that's kind of how it worked. So when we ended up duplicating Hox genes, we ended up locking into particular body plans and types. And that's why we have the Cambrian explosion, as they call it. So anyway, let's see. Okay, I think I've said enough for now. Have I made you sick? Calling it just cells either trivially true of all, therefore irrelevant, or smuggling in an assumption it's a real organism, which is empirically wrong.
I think AI here is just having a discussion that nobody else is having. I mean, we're not really talking about the personhood of the embryo. We're talking about evolution and what kind of insights we can get through the development process.
Yeah, but I did want to emphasize that this thing about personhood, right? When you think back to when you were just that little tiny inner cell mass, and you had undifferentiated cells, and even part of that was going to end up becoming the amniotic cavity or amniotic membrane. A lot of you, like, what were you at that point? What would be your personhood at that point? And where exactly is the soul generated, injected? So those are questions I have for some Christians. Okay, Ediacaran, biota, soft body, mostly simple organs. The Cambrian explosion is one of the biggest unsolved puzzles. Yeah, but it's not really that much of a puzzle. I just explained how it happened. Basically, you end up with these hierarchies of differentiation going from the outside of the onion all the way in. And the more complicated those systems become on the inside, the more locked in you get. In the Cambrian and the Ediacaran, they were a lot simpler. These animals were a lot simpler. And the other thing about the Cambrian and Ediacaran is that bone, we had cartilage development and we had calcium storage, but we didn't have skeletons yet. That was coming later. And when skeletons came, so did fossils. And it's a lot easier to get a fossil record.
I asked Copilot just to get a idea of how accurate that statement is with respect to the current consensus of evolutionary biology. The statement is not accurate. The Ediacaran Cambrian transition is scientifically active and complex, but it is not considered one of the biggest unsolved mysteries in evolutionary biology. The broad outlines of what happened and why are well understood, even though details remain debated. Below is a clear breakdown grounded in the current consensus and recent literature. I'm just going to give the the big bullet points and not go into
Yeah.
detail. The transition is well studied, not a major mystery. The fossil record is improving, not mysteriously empty. The Ediacara and biota are not simply soft-bodied, simple organisms. The claim that this is one of the biggest unsolved puzzles is inaccurate. So that is definitely fodder for future research, in personal, individual research study.
Yeah, this one transition from single cell life to multicellular organisms. We know a tremendous amount about that right now, and that's one of the things that I study is the extracellular membranes and the stickiness of cells, how cell surfaces stick together. All of those mutations happened in preceding multicellularity. But once those things happened, we ended up with something called cadherins. And cadherins are really interesting because one cadherin will stick to its own type, right? It'll stick to itself, that sort of thing. And then a very slight mutation in that cadherin will mean that it can no longer stick to that cell type, but now it'll stick to another cell type, right? So we end up with cell sorting. And cell sorting was the beginning of organogenesis. That was the beginning of how organs evolved, because cells would sort into two things. And those cadherins and other proteins like them with very small mutations can end up differentiating a cell type so that one cell type will stick to its own kind. Some cell types want to stick to another kind. And then we have a difference in tissue layers, right? So now we have this sharp division. And it's all of that mechanics of the cell stickiness that ends up creating the jump. Single cell life for billions of years before we ended up with the mutations that created cell membrane stickiness, basically the exuding extracellular matrix and all of that. Those steps aren't.
Yeah. Once again, I put that
statement aren't missing.
Yeah, I put that statement in Copilot, asked it to do the same kind of analysis. The statement is inaccurate. The intermediary states between single cell life and Ediacara multicellular organisms are not missing. They are extensively documented across multiple lineages, and the evolutionary multicellularity is one of the best understood major transitions in evolutionary biology.
Yeah.
Multicellularity evolved many times, not once. The intermediary steps are well documented, and it gives a list of different examples. The Ediacara does not represent the first multicellular life. What is debated is how many times complex multicellularity evolved in animals, the ecological triggers for Ediacara diversification, the developmental genetic innovations involved.
Yeah.
So I think that would be an interesting course of study to engage in for all parties involved.
Something to note at the top is the Ediacara and the Cambrian are lines that we put on paper. It was a continuous timeline, and there was a continuous lineage of organisms. And right around the Cambrian is where we ended up with sufficient gene complexity or chromosome complexity and duplication that we locked into several body plans that ended up out eating and out competing all the others. And that's why we ended up with the body plans we did. But in the beginning, from Ediacara to Cambrian, we had very, very simple developmental pathways. And those simple pathways could, with like a butterfly wing flapping, could change from one body plan to another. And one of the things that I noted at the beginning of this is that in the mouse, at that morula stage, there are a different number of cells than there are in humans. And that ends up changing the entire course of that organism. And those things could happen very easily if the organism had stopped developing shortly after. Okay. If we had a viable organism like a sponge or something simple that just sat on the seafloor filtering water or whatever, it would be a lot easier for that to mutate into an entirely different body plan or to turn into two body plans. And that's what happened back then during the Cambrian. And we know a tremendous amount about that cell stickiness right now. And we have all kinds of really neat, if you look at the NCBI genome and you look at these sticky proteins, cell sticky proteins like cat herons, you'll see where they duplicated and mutated and duplicated and mutated and did this, like a half a dozen times, and ended up creating this massive family of proteins that ended up with the complex differentiated life that we have today, which we did not have in the Cambrian. Now, I want to say that again, in the Cambrian, the organism types were simple enough that basically a little bit of change in regulation could have created a very different looking organism. And I think when you look into, like we're always stuck looking at mammals. When you look into the biology, marine biology, you find so many examples of unbelievable complexity that's developing and has developed over and over because the body plans are simpler and the regulatory networks are simpler. Anyway.
We've been going over this for over an hour, so we might want to break it off and go about our day.
I want to go about my day. I got things to do, man. I got I got to talk to you about my farming endeavor. I'm having I'm at a great existential crisis, but I want to acknowledge what is it, AI flyer? Ediacaran, okay, quantum flagellates, and colonial share some genes that animals would know. We share a lot of genes. Observed evidence demonstrated how similar. Yeah, we're not going to observe evidence of one thing developing into another. We do it with biological, genetic pathways and molecular biology. We understand a great deal about that. And if we had the time and money to do the work, we could do it in one step at a time, and we could show you how it works. So, no, they didn't appear all that abruptly, but yeah, I guess, yeah, it was, in geological time, it seemed to happen real fast. It's just that there were all these different possibilities back then. Anyway.
Maybe if I went to a different login and a different AI,
yeah,
the responses might be different. But I put the complex animals with eyes, nervous systems statement in Copilot, asked for the same kind of evaluation according to the consensus of evolutionary scientists. The statement is inaccurate. Complex animals in the Cambrian do not appear abruptly. And there are well-documented evolutionary pathways, both fossil and molecular, linking them to the scientific consensus is that the Cambrian diversification was rapid in geological terms, but not sudden and not disconnected from earlier evolution. Below is a clear evidence-based breakdown, blah, blah, blah. So maybe, because of, my history with this particular instance of Copilot, a different instance on a different computer of Copilot might, or some other LLM might produce different answers. Otherwise, we might have to get somebody who is a evolutionary scientist who studies the Precambrian Ediacaran in here to see what they say about it. But this stuff, AI Flyer, I think you're just putting out misinformation, disinformation because of a dogmatic commitment.
Yeah.
But we're not saying the evidence is in the future. We're saying there's lots of evidence that it's not happening like you characterize it, and that we have good reason that in the future we'll have. No, you're just mischaracterizing things. AI Flyer is my opinion.
Here is
that's my opinion, man.
Yeah. Yeah, it's just the thing is that these okay, Ediacaran, right? Complex animal body plans did not just develop in the Cambrian; they were simpler in the Cambrian. That is the point you have to understand. They weren't the same. Thumping, thumping, Scott. You're thumping a lot, man. Thumping a
Yeah,
lot.
I need the phone.
Okay. Here's this is the source book for a lot of the stuff that I showed you and a lot of the illustrations. Basically, this is Larson's. Oh, let me see here. This is Larson's Human Embryology, 6th edition, 2021. This book cost me a pretty penny. I think about $200 for this one. Let's see here. Let me go down to where this basically, yeah. So what I'm doing here is I'm doing a lot of highlighting. And the reason I'm doing a lot of highlighting is because this ends up, I have something called highlights, which takes a PDF document and it'll break those into one-liners. And then I can stick them in my freeform thing and that's basically how I study. I separate, like I do all the terms in green and other things in blue. And basically I can turn this into a wonderful note-taking device. But let's see, here's all the pictures. Where are we here? And that's about as far as I am. I'm on page 133 right now, heading into the next page. Anyway, so we'll do a little bit more of this. And the other thing is I have some other developmental embryology books, really good ones that they use to teach this in college level. And they deal with other organisms. And that's the interesting place where you can see the differences in these developmental pathways, in these embryological pathways. And you can start to visualize how very simple changes in regulatory parts of the genome can create different body plans and different things. So let's see. What do we got, Scott? Yeah, we're going to get out of here now. Yeah,
Thanks for stopping by, Ocean Helios. You came in kind of towards the end of things. It would have been great to have you closer to the beginning, but
yep.
Always great to have you.
Yep. Yeah. Eyes, antennas, legs, and such have been reused throughout the tree of life. Yeah, the reuse is part of the problem. Like I say, you get locked into, like mammals, the mammal type, and you're never going to go backwards. There's too much to do to go backwards. Anyway, okay, we're going to end this, Scott. I'm done. You ready?
Yeah, I'm done too.
Oh, well, you should have said something. Okay, going to end this, guys. Now, AI, whatever, I'd love to talk to you in comments. Please, let's engage in comments so we can go through some of this and try to straighten out the differences that we have.
Well, at least you didn't get a tag argument.
Yeah. Laborious Cretin, it's always good to see you over there. Even though sometimes I don't understand half of what you say, but I understand half of it. So love the input. Okay. Goodbye, crew world. We're out of here.